Okay, so good morning, everyone. So, um, I would like to welcome everyone to this lecture series. So, for this lecture series, this is so good morning, everyone. So, um, I would like to welcome everyone.
Mute po ng Facebook live stream.
Okay, sorry. This lecture series is organized by the Philippine Council for Health Research and Development, Balik Scientist Program, Department of Science and Technology, and the Department of Chemical Engineering, University of the Philippines, Diliman. So this is the lecture series about microfluidics, expanding the frontiers of medicine. So this is a lecture series on the biomedical applications of microfluidics and nanofluidics. We are on the second of the third of the three lecture series. And before we start, I would like to introduce our speaker for today. So, our speaker is a multidisciplinary scientist with strong background in applied mathematics, chemical and biomedical engineering, biophysics, and medical sciences. She's a UP alumna who holds several master's and doctoral degree with education and trainings from Harvard, MIT, Oxford, and Cambridge universities. She is presently a Global Health Delivery Scholar at Harvard Medical School, an AAUW Fellow at Massachusetts General Hospital and Beth Israel Deaconess Medical Center, and a Schlumberger Fellow Alumna at Cambridge University. So without further ado, so let's start the webinar. So we have our speaker, Dr. Jhalique Jane R. Fojas.
Hello, thank you, Sir July. Good morning, everyone. Welcome back to some of you. I'm quite excited to discuss this particular topic because I've always imagined building a PlantChip integrated with, organs on a chip or even systems on a chip as a child before I even heard of the term microfluidics. It would have been a great test run for engineering myself to photosynthesize, Don't you want to simply get close to the window, spread your arms and feet out of the sun, especially during this pandemic, where everything is some sort of a survival of the fittest run. Or likewise, even as a child, I also dream of engineering some humans, mostly bully kids, to glow, basically tagging them with GFP and DAPI, for example. And all these dreams come back to life during this pandemic, where one has to have multiple roles to play. I'm sure you do at your own homes, and hardly had the time to eat, let alone, play Pac-Man inside the supermarkets. I've seen this meme. And of course, it's really hard to also starve and not eat during pandemic, especially while watching. So I've seen the news this morning while watching corrupt politicians steal from our Kabanang Bayan. So I was thinking we need to fluorescently tag them, develop a greed evil tagging fluorescent protein. So this is my challenge to fellow millennials out there and the generations after us to identify the threshold for selfishness or greed and find the biomarker so we can tag the likes of these corrupt politicians prior election time, save this planet of space and oxygen. So this is what happened when you watch the news early morning before giving a lecture. You see all the misdealings of national politics. Anyway, moving on to the science at hand. PlantChips, what are they? And why develop them when you can throw or spit seeds into your neighbor's backyard? We don't have a backyard. And they grow anyway. Or, for example, leave mango seeds in the sink. So I was supposed to share the screen, but let's see if my connection would allow this. There seems to be a lag. If you can see the presentation. Not sure if is everyone or am I on mute? Can people see the screen? Give me a shout out if you can actually see the screen properly. So moving plants and chips. So what are microfluidic chips in plant study? Thank you, Sir Champong. My internet connection is really awful. I think the downfall of the Philippine economics is because of our sluggish internet connection. Anyway, so too much of current events early this morning. Anyway, moving on. So like what I was saying, why do we have to build plant and chips? What are they for? When, conventional methods of plant cell analysis and plant cell examination techniques rely on growing plant cells in soil pots. So for those plantitas out there, or agarose plates for those biologists, followed by the screening of the plant phenotypes in traditional greenhouses, nurseries, and growth chambers. So this is a highly expensive type of system. These methods are obviously very costly. They need large number of experiments. They also suffer from low spatial resolution. So, the potential data loss during the observation of plant phenotypic changes because of low sequential resolution. And of course, the disorderly growth behaviors of plant cells or plant organs or the entire plant, the lack of ability to locally and accurately manipulate the plant cells, the plant, and even the environment. So all of this are the downside of conventional methods of plant cell analysis or plant cell examination techniques. And like I mentioned before, for those of you who attended my previous lecture, and I didn't put in a comma, and were listening, microfluidic platforms take advantage of miniaturization for handling small volumes and providing a closed environment, more like controlled environment, with the purpose of in vitro single cell analysis and characterization of cell response to external cues or stimulus. Next slide, please. So these platforms have shown their ability for high throughput cellular analysis with increased accuracy of experiments. It's more economical to reduce cost and experimental times. Versatility in design, you can design very simple microfluidic devices to a highly complicated one. And also the ability for large scale and combinatorial screening. So you can screen hundreds or even thousands of compounds, for example, in a microfluidic device. And lastly, integration with other miniaturized sensors, so that you would probably have read in MEMS, for example. So what is exactly plant in chip? So like organs on a chip. So it's a microfluidic device. It mimics the growth environment of a particular section of your plant or an organelle plant or an entire plant, for example. And it should have the ability to regulate parameters. So this includes concentration gradients of, the nutrients supplied to the plant, fluid flow. So how does the transport inside the plant look like, patterning as well. So do you pattern the roots, the shoots, etc. Availability of in situ analysis, can you do an assay inside it? And then elimination of composition change in biological system and time lapse between sampling and analysis. And you have also the flexibility and mobility. You cannot simply move your farm or your backyard from one place to another. I barely had the time to move my sampaguita pot from one side of the condo unit to the other. So with microfluidics, all of these constraints could be addressed. Next slide, please. Now, let's look into several types or the common types of planting chips or microfluidic platforms for plant cells analysis. Here you have, let me try to draw that. So here you have the RootChip design. So with the RootChip design, the seeds are actually manually located inside plastic tips. So you have plastic tips over here. The roots elongate through the agar field. So you have the agar field chamber, conical cylinders. Then it reaches the growth chamber and monitored within the growth chamber in horizontal plane. So you have your microscope here at the bottom, so you can observe what's going on with your roots or with how the root elongates and what rate it elongates, etc. Another plant in chips type is your RootArray. So the purpose of designing RootArray is to study spatiotemporal gene expression dynamics in plant cells. Roots and shoots grow in liquid chambers. So you have both the liquid chamber and your gaseous chamber. So that's a liquid chamber and gaseous chamber respectively. So for roots and shoots. And then the chip is sealed by two cover glass. So you have here two glass cover slips, for example, or you can use cover glass as well at both top and bottom surface. The advantage of tips is that cover slips is that it's very thin. So when you're imaging, you get a better resolution. The roots then can be monitored in both vertical and horizontal positions. So you can monitor both this aspect and this aspect, primarily because if you're using a translucent material like PDMS, for example. And then you have PlantChip. So the schematic design of your PlantChip for high throughput plant phenotyping is shown here in image C. Seeds are hydrodynamically, so these are your seeds, they are hydrodynamically trapped at the entrance of the funnels. The roots and shoots are then monitored in their vertical positions. And then another common type of planting chips is that of the TipChip. So the overall design of TipChip shown here in your image D with serially arranged growth microchannels. So you have growth microchannels serially arranged. The pollen tubes are automatically trapped at the entrance of your microchannels. So you have your inlet here and you push your pollen tubes in there, in these microchannels. The growth of pollen tubes are then monitored along the growth microchannels in the horizontal plane. And then you can monitor the growth of that here. What are the other applications of planting chips apart from the 4 common types that I've mentioned? So here you can see in the table the ranges of applications of planting chips that have been explored out there by current researchers. There are many indications that the microfluidic technology will actually reshape the plant science and agri-industry for plant diseases treatment, even agricultural modification to change or adapt in environmental conditions, with climate change, with sudden rain pouring. So those kinds of things, you cannot control that if you planted them out there in the fields, but in microfluidics, you can easily emulate this and you can even control the conditions. So on top of that, we have water shortages because, like I mentioned a while ago, climate change. And then the increasing demands for food and supplements. So, in the Philippines, we have this golden rice. Unfortunately, it didn't take off. But anyway, so supplements and even medical drugs. So in this particular lecture, our focus is more like on biomedical sciences, medical applications of plantain chips. In this table, you will read some of the major applications of PIC that have been explored, from environmental system monitoring to in vitro investigation of plant cells, root shoots, and also segments to transfection studies like the ones mentioned at the bottom, where they measure even cell divisions for cell damage, cell division studies. now let's take a look at the different common types of planting chips and the current studies that have been done with regards to the development of these chips and their applications. Next slide, please. And so the first common type of planting chip is RootChip. Next slide, please. So, roots act as the interface between plants and soil. They take up water and nutrients, respond to changing environmental conditions such as water availability, nutrient concentration. You have the salinity, pH, and the secrete exudates, all of this. To understand the molecular basis of acclimatization or equilibrium processes within the living root tissue, technical advances are needed to perform assays at cellular resolution, while of course varying the root environment or changing certain conditions or external factors around the root growth. Important performance parameters, which include the speed and reproducibility with which you change this environment or you manipulate this environment, can be made easy or even reversible using microfluidics. So the stability of environmental conditions over time, and the ability to perform assays in multiplex to improve basically your experimental efficiency and the achievement of results for a particular application that you're interested in. So in RootChip, here you can see a RootChip microfluidic device wherein they integrate parallel root culturing. So you have here the roots of your plants. So here they integrate the parallel root culturing with temporal and content controlled perfusion for roots on a live imaging platform. So essentially you're looking at how the roots grow and respond to particular stimulus live. So you have your imaging system at the bottom of your chip while the root is actually growing. The utility of RootChip is demonstrated in this particular study with Arabidopsis roots expressing a genetically encoded fluorescence sensor for your glucose and galactose based on FRET, or what you call forced resonance energy transfer or fluorescence resonance energy transfer, or sometimes it's even called resonance energy transfer or simply EET, or your electronic energy transfer. So FRET is a mechanism describing the energy transfer between two light-sensitive molecules. So in terms of your plants, you have the chromophores, for example. So such FRET sensors can be incorporated in your microfluidic chip to allow for non-invasive real-time detection of metabolite levels and influxes or fluxes in living tissues or in plants, in particular in the roots. And this subcellular resolution can be achieved by targeting sensors with specific signaling to different cellular compartments. So this is exactly what they did here. So they coupled, they have a planting or root chip microfluidic device and then coupled it with a sensor to detect the changes in the fluxes of the changes in the concentration, for example. Now, we can look at further the results for this chip and even the design for this chip in the next slide. So here you have an image of what the root chip looks like. So the root chip is this one. And the image A, so this image shows you basically a PDMS chip. So this is actually a PDMS chip with 8 mounted live plants. So those plants with the control and flow channels of the chip. So these are actually your flow channels filled with red and blue dye. So just for the purpose, I think they only color this with red and blue dye for the purpose of taking an image, a good image to show you, excuse me, to show you the flow. And then you have here image B, which is essentially just the top view of the 8 plants in conical cylinders. So this conical cylinders, actually, this looks like more of a, tip pipette tips that you can just cut out. I've done it before. You need to be a bit more innovative when it comes to this. So these are conical cylinders filled with agar and mounted into the chip with the arrow indicating the growth direction. So this arrow just indicates that the roots are growing downward. The image on the top right, so this one, shows the bottom view of the microchannels. So basically what you cannot see in this photo is this one. And these microchannels containing roots, your roots, of seedlings 7 days after germination. So it's quite fascinating to see how their roots are actually growing inside the channels. And then you have the lower right image, which shows essentially the color map analysis of your acceptor donor intensity ratios while the roots are growing, with green and red shaded bars that indicate basically your perfusion with sugars. And then you see that the phenotypes of your root tip regions are prolonged perfusion with sugar-free. So you have here the sugar-free, and then you also have here in the middle your glucose, and then glucose containing medium, and then you have the galactose containing medium. Oops, I couldn't. Why can I not write that? Okay. So that's basically what they did in this type of experiment. And of course, you can have several applications apart from monitoring the growth of the root chips and from changing the type of media. So you can also change, you can also change you can also test other chromophores in this particular device if you wanted to. But in this particular study, they only look into the glucose and galactose. And another microfluidic device involving plants is the RootArray. So I mentioned this a while ago in the beginning of the lecture. Next slide, please. RootArray. So why do we have to develop RootArray or this type of microfluidic device? Go back to the first, go back to the previous slide, please. So another major challenge in biology today is mapping genotype to phenotype. A key aspect of the mapping function is actually gene regulation. And it is increasingly evident that we need to capture the dynamics of gene expression to understand the complexity of its regulation. And this particular study is very challenging in multicellular organisms, in which spatial as well as temporal aspects of gene expression need to be monitored and for quite some time. One method to address this issue is through time series expression analysis using microarrays or sequencing, which in general, if you do it in a conventional way, is performed on whole organisms or the entire organs, with only very few methods developed to actually isolate specific cell types or specific developmental stage. The effort of then extracting specific cell populations and the cost of profiling them at different time points are prohibiting, especially in a setting like the Philippines. It's very costly to do all this. An alternative method would be to use fluorescent or light emitting reporters and to image over time at the resolution of individual cell types and inside a very small device so that we conserve the reagents, for example. And this is what has been done in this particular research by Bush and his colleagues. In order for them to fully describe gene expression dynamics, they need to quantitatively capture expression in individual cells over time. So as you can see here, different time points. Automated systems for acquiring and analyzing real-time images are also needed to obtain, unbiased data across many samples and conditions. So, in biology, you always have to look into the reproducibility. So you have a lot of samples, actually. Sometimes you need more than 10 samples to actually get a statistically significant result, and even hundreds. Here they develop a microfluidic device in which 64 Arabidopsis thaliana seedlings can be grown and their roots could be imaged by a confocal microscopy over several days without manual intervention. So essentially, I've done this in the past with, but it's not a plant on a chip, but rather as an eye on a chip, wherein we'll look into the optical neurons. So the growth of the synapses over time. So we essentially just put the device under a confocal microscope and then program the microscope to take images at a particular time. And we did that for a week. So much data. And that's essentially what they did here, but they're dealing with roots. In this particular figure, they showed the progression of expression change in WOX5 reporter lines. So what is this WOX5? So WOX5, the maintenance of the stem cells depends on the transcription factor called WOX5, which is Waschel-related homeobax5, which is specifically expressed in the group of slowly dividing cells named the quiescent center or year 2C. So this is what they monitored here. They monitored the progression of expression of WOX5 in various growth conditions, including you have the standard medium. So this is all standard medium. Oh, my pen is, let me change the color. So this, for example, all of this, A to E, are standard medium. And then they also look at what happens to the same, similar growth cells when you have a sulfur deficient medium. And that's what you have here in your F to J. And then they also monitored the changes in the growth if you have low pH. So you have that here. And they show in this particular results the insets for your, insets or your magnified portions indicated by your rectangular box here, just to show you the low resolution images of the roots at the last time point. And so basically they monitored what happens to the growth of the roots at various conditions wherein they have different type of medium and different pH of medium. Next slide, please. Now let's look at the device itself. So in this slide, you can see the image of the RootArray with 64 wells and its cross section. So this is essentially your cross section. With the dimensions of the design, so you have there, it's pretty big actually, it's 2.59 millimeters, and then you have 300 microns. So relatively this type of RootArray could be actually produced with 3D printing rather than the typical, photolithography. The wells connect both the chambers, and it is allowing the roots to actually grow into the liquid chamber and the shoots to emerge essentially into your emerge into your gaseous chamber. The upper and lower edges of this illustration indicate cover slips. So they have cover slips in here that they use. And then for image C and D, you have here the growing of your roots as image over several hours. In this case, the lower image is after 21 hours. And then at the bottom, you can see an image showing the maximum projection of your stitched 3D high resolution image. So the black lines that you can see indicate the computed border and the medial axis of the root. And then the image here is the extracted root, okay, with sampling points used in the computational. So you have sampling points. Computational straightening mark as your red line. So those are sampling points. Then they also made a computationally straightened medial section of the same root. And of course, the 3D plot of the identified root, its medial section here, And then you have the red portions. Basically, this is a map. So it's a heat map wherein you have the red portions, which are close to the surface, and your blue portions are farther away. And then, of course, you have the medial axis, which is in black. So what do they do essentially? So they calculated the growth rate, counted cells expressing a cell cycle marker indicative of the division, and measured the dimensions of the different developmental zones. So they determined that the average growth rate of this particular root is 70 micrometers per hour under standard medium conditions in the first 24 hours. And they also determined that under this standard condition, there was a lower, it has a lower reported growth rate compared to your older plants in vertical agar plates. Although they saw that there is no evident cell death in slowing the growth of the roots, they indicate also that the slower growth root was not adversely affecting the plant. So here they observed that the growth of the root is slower, but consequentially, it doesn't really affect the plants adversely. So here, it's advantageous to actually grow it slowly because you can monitor the morphological changes, as well as if you integrate it with some assay, the chemical changes in the growth roots. And so another application of microfluidic device in plants is PlantChip. Let me just erase all these things, all my doodles. So this other PlantChip platform studies the plant uptake of essential nutrients from soils. So in this particular PlantChip. So we all know that nutrient stress is one of the major factors that cause unhealthy plant growth. So for example, my sampaguita almost died 2 days ago because I forgot to essentially give it some water. So real-time monitoring of nutrient availability during plant growth is, of course, critical to regulate nutrient levels for reducing, let's say, fertilizer or manipulating certain plant products. So if you want, let's say, our golden rice, if you want something with high vitamin A, then you could manipulate the nutrient source or even the plant itself for that. So two of the most important nutrients from the soils, or nutrients that would be the sources of nitrogen and phosphorus in plants. So nitrates is the main source of nitrogen. It is an important macronutrient for crop production in agriculture. And understanding this, of course, understanding the nitrogen use efficiency will help the timely management of nitrate input to agricultural soils or systems. So we wanted to understand when would the plant need more nitrates in its growth span. Additionally, nitrate also serves as a potent signal to alter plant metabolism and control gene expression in plants. And then I've mentioned a while ago, phosphorus is also another important component. So phosphorus is a catalyst in the conversion of numerous key biochemical reactions in plants. Exists mostly as a phosphate, or in this case, in this particular study, I think they look into the dihydrogen phosphate and hydrogen phosphate instead of just, phosphorus. Here in this image, they report on the development of a miniature plant growth device integrated with ion-specific nutrient sensors for real-time monitoring of your phosphorus and nitrogen uptake of plants. The device consists of your PMMA. So we've mentioned from the last lecture that you could use various materials in producing your microfluidic device. It's just so happened that everyone's using PDMS, but essentially you can also use PMMA or your polymetal methacrylate. So here they use PMMA chamber filled with growth medium and ion-specific nutrient sensors formed on the bottom surface of your chambers. And a plastic mesh in place on top of the sensors to avoid, of course, the direct contact. So to avoid the direct contact of your roots with the sensors. So let's try to look into the results of the study that they did in this PlantChip. They quantify this uptake using, sorry, excuse me, let me just get some coffee. I need to lubricate my, otherwise I'll run out of place. So they then quantify this uptake using the calibrated sensors that measure your dihydrogen phosphate and nitrate in the growth medium. So the dihydrogen phosphate and nitrate concentrations in the medium were monitored over 5 successive days, as shown in the figure. So if you can see this, you could identify how long they measure the uptake. In addition, as the growth chamber was replenished with, so these are the growth chambers, they replenish that with medium once every 5 days, over 15 days. So you can imagine that it's not just day 1 to day 5 that they monitored, but they actually monitored day 1 to 15. So the rice plants were found to routinely uptake your dihydrogen phosphate and nitrate. So you can see that, this is obvious here. Like how the increase and or decrease in the uptake. And then you have here showing that the plants actually were shown to uptake less amount of dihydrogen phosphate than that of your nitrate, because essentially a larger amount of nitrogen was required during seedling. So this is one of the like amazing findings. Most of our farmers probably know this already, but looking at it in the microfluidic chips, and you can see why it does make sense that actually nitrate, you consume more nitrate during the seedling process compared to dihydrogen phosphate or your phosphorus. And you could do other things with this particular chip, not just monitor, the phosphorus or nitrogen uptake, but you could also check on other nutrients needed by the plants, or even just the water uptake of the plant, for example. Now, another common type of your PlantChip is what you call the TipChip. So this device was developed based on microfluidic and microelectromechanical systems, or your MEMS technology. The large-scale phenotyping of tip growing cells, such as pollen tubes, has been limited to crude parameters like germination percentage and velocity of growth, like the ones that I've shown you, the past studies that I've shown you, are mostly on germination and velocity of growth, or the effects of changing medium parameters with respect to the root growth. And here what they did, the authors developed an experimental platform that enable efficient and high throughput execution of your more sophisticated assays. The device here allows you to, allows positioning actually of your pollen, and pollen grains, in this example is pollen grains. You can also use fungal spores at the entrance of your serially arranged micro channels equipped with microscopic experimental setup. So like I mentioned a while ago, what the advantage of microfluidic chip is that it's because it's very tiny, you could design it, it's translucent. You can always couple this with high resolution microscopy, or not even high resolution microscopy. Of course, if you wanted something like a nanometer or a subcellular analysis, then you have to use high resolution microscopy. But for typical laboratory setup, you can just use confocal microscope, for example. Here, the tip growing cells like pollen tubes, filamentous yeast, or fungal hyphae may be exposed to chemical gradients, microstructural features. So you have here microstructural features, and then you have you can also expose that to different chemical agents like or gradients like the ones that I've discussed when we were talking about organs and chips. And then you can integrate biosensors or directional triggers with the modular microchannel. So let's try, it's more like trying to see if they would grow closer to a certain high level or high concentration molecules compared to low concentration molecules. And then all this, so they look at the tip growing cells. They also focus on their growth activity onto a single site on their cellular surface, and how they regulate growth dynamics by essentially exerting temporal and spatial control. And this is what they did in this research. Here you can see basically the overall design of their TipChip. So that the image of the TipChip is shown in the PDMS layer attached to cover slip, and you have the inlet outlet. So it's very simple. If you could see this, okay, I'm in trouble with my pen. It's a very simple design, really. You have inlet, and then you have several outlets, and then you have the microchannels here. So you could change different designs for your chamber. And then the general design principle of this microfluidic network in the TipChip is that the pollen suspension is injected through your inlet, and then the pollen grain moved through the distribution chambers. So you have pollen grain moving to your distribution chamber, and they either become trapped at the entrance of the microchannels or are evacuated through the distribution chamber outlet. So you can particularly sort out what gets trapped and what's not. And then here you also show rectangular distribution channels. For example, here, this is a rectangular distribution channel, and it's a simple device. Then you can do different geometries if you want to. So changes in the geometries, you can play around with this. And here you also have fluid flow simulations in the chambers. Next, in the next slide, you will have here a more obvious application, because now you can see the growth, you can see the tip growing of the plant. So, tip growing plant and fungal cells have been widely used as model systems for cellular morphogenesis in walled cells. So the growth in walled cells occurs through turgor-driven expansion of the cell wall and simultaneous assembly of new cell wall material. So tip growth is characterized by, of course, spatial confinement of the cellular expansion process to the very end of the cell. And the resulting structure is a cylindrical protuberance, which is quite perfect if you look at this. And sorry, just something. So among the hallmarks of these tip growing cells would be the speed of the growth and the ability to control the direction of the growth. So it reminds me so much of neurons, really, to be able to see the speed of how it's growing and then the direction as to which side is has less resistance for it to grow in that direction. And as you can see in this particular work, they studied how kink and so you have kink and serpentine, excuse me, to prevent pushback and test the endogenous memory. So the pollen grain of a pollen tube growing through a microchannel against a mechanical obstacle is pushed back, and the pollen tube is thus unable to exert a mechanical force against the obstacle. Hopefully this is not the same in life, that when you encounter an obstacle, you simply just cannot grow. So a pollen tube here in image B, you have a pollen tube growing through a microchannel with inverse fluid flow. So despite the current, the inverse flow, it still managed to grow, but of course not as good as those without obstacle or with flow, which is in the same direction as the growth of the pollen tube. And here you introduce, in image C, you introduce a certain kink, incorporation of the kink in the microchannel architecture. And this enables the pollen tube to actually remain in place, but you can see it curves. So mechanical obstacles or an inverse fluid flow encountered after passing this kink do not cause displacement of the grain. So this is what they found out. It has allowed the tube to exert a pushing force. And in image D, for example, you can see here the actual pollen grain of, I think this is camellia pollen tube, growing through a microchannel against a mechanical obstacle, which, tried to push it back, but still it continues to grow. And here in E, the same camellia pollen tube growing through a kink microchannel. So look how flexible it is, that they could actually go around the kink and still grow. So in a proof of concept experimental test, pollen tubes were exposed to a gradient between 8 and 12% sucrose. So this is an additional findings in this study, wherein they exposed the pollen tubes to a gradient between 8 to 12% sucrose in growth medium. And they realized that they turn towards higher concentrations. So this is what I'm talking about, that will they like less sucrose or will they tend to grow in more sucrose? And the TipChip, this particular device, actually allows high resolution imaging using Nomarsky optics, as well as confocal laser scanning microscope. So this is the advantage of developing microfluidic devices or putting your plant segments or pollen tubes inside your microfluidic chips. So because it's translucent, you can observe real time what's happening in your system. And this is exactly what they did here. You can see here the confocal laser microscopy images. So you can see the images H, I, and J, for example. And they observe that the higher the concentration of sucrose, the pollen tubes tend to grow towards there. And who doesn't like high sucrose, right? Even I would thrive in, with more chocolates and sugar, lots of sugar. Anyway, so moving on, I will also be discussing integrated. Okay, let me erase all the doodles first. So moving on, let's discuss also integrated planting chips. So planting chips are not restricted to simply determining the growth of particular roots, how fast it is, or what particular solutions will it grow faster or grow towards to. There are also integrated planting chips wherein you look into combining microfluidic device or simple PDMS chip with sensors, for examples. And these other integrated Pics were developed for a more complex applications. So for example, in this slide, we're looking at chip developed to measure antioxidants in plants. So it's gearing towards more of the biomedical applications like antioxidants, for example. So antioxidants are important substances that can fight the deterioration of free radicals, your OX. And can easily oxidize when exposed to light. There are many methods to measure the antioxidant activity in a biological sample. For example, you can use your DPPH. So that's I think that's 2,2-diphenyl 1-picryl hydroxyl. So any chemists here in the audience? And so you can use DPPH antioxidant activity test, for example, which is one of the simplest methods that are available. But despite this simplicity, this organic solvent that has used to dilute DPPH is easily evaporated or degraded with respect to light exposure in time. So you have that blockage there of it being exposed quite easily and then it degrades immediately. So thus, you need a device that could conveniently control the exposure to light. And to overcome this issue, in this particular study, they use a rapid enclosed system for antioxidant activity. They introduced this lab-on-a-chip method that essentially integrates the PPH antioxidant activity test on a microfluidic compact disc. And so the disc is this. You can see that. So you can see several channels in there. You can see 3 channels actually. So the initial experiment is your A. So you have here basically time 0. And the speed is 0. And then you have the plant extract solution. It started to flow in your capillary. Oops, I did something. Sorry about that. So you have the plant extract solution started flowing to your capillary and capillary valve, and then speed has been increased from 0 to 300 RPM. So this is time 0. The speed here is 0. Oops. Speed here is 0. Sorry. Velocity. Keep thinking about the speed of light. So anyway, speed is 0. And then you have here the plant extract solution started to flow in your B. And here you start the speed from 0 to 300 rates per minute. And you can see that slowly it's moving to your capillary valve. And then the plant extract chambers have been emptied at about 300 RPM. So the TPPH solution will then flow out from its chambers to the capillary valve to meet the solution from over the plant extract solution. And then speed has been increased so slowly from 300 to 800 RPM. So this is what they do. So they try to mix it slowly, the plant extract moving to your capillary valve and then your DPPH solution. And all solutions will then mix properly. So you can see that here from figures A to F, you can see how the movement of your solutions until you reach a properly mixed chamber. And now what do they do? Why are they developing this type of device? So let's look at the next slide. Sorry, let me erase that again. Okay. So to study the developmental process in plants, the sample must often be, dissected to access the area of the plant where the tissue is developing and differentiating. So I see. So here is another study on that. And in this particular work, they look into the development and differentiation of area of a plant or a tissue. Because taking time points requires serial specimens. So when you're looking into different time points for a particular system, sometimes you have to, so just for example, in a solution. So if you're looking at the concentration of, wine, for example, wine. So the concentration of, I just saw some, anyway, so I just saw some wine photo here. So a message comes in. Hopefully you didn't see that in my shared screen. A message came in and there was a wine photo. So anyway, so particular wine. So if you're trying to sample and look at the different ethanol concentration of a wine from a glass of wine that you're fermenting, you take in a particular amount every time until that amount depreciates over time, right? Until you have like one glass of wine completely used up. So here, what they wanted to do is to look at the how does the changes in specimens. And if you're doing conventional system, you would essentially get one specimen, measure it, and then essentially use it all up in one time point, and then you have to look into another specimen. But that specimen is quite different from the previous ones. So this particular integrated PlantChip did solve that or address that problem, wherein they're taking time points serial specimens, and they're looking at the developmental process within a single specimen. So here they describe the design and utilization of PDMS, microfluidic chambers, for long-term imaging of the development of a moss. So this one is a moss, Fiscometerella patens. It's a simple body plan, and it has excellent cytology. That's why probably it garnered interest as a model organism in most evolution of developmental processes in plants on cellular and subcellular levels. Moreover, in this particular study, they also look at the ability to efficiently edit the genome via homologous recombination. So they emphasize how amenable microfluidic chambers are for confocal microscopy. So like I mentioned a gazillion times, not really gazillion, but a couple of times, the microfluidic chambers have advantages in its flexibility and portability, that you could couple it with several devices. So essentially, in this study, they coupled the PlantChips to confocal microscope. In the figure at the top, you see the YFP targeted to the Golgi, revealing a tip-focused accumulation of your Golgi dictyosomes, whilst your M-cherry, so essentially, to put it in layman's term, you tag different parts of your plant system with a certain fluorescent dye. Like, for example, here, they use YFP for the Golgi. They use M-cherry to target mitochondria, and then they use GFP to target peroxisomes to look into, and also GFP gas for fusion targeted to the nucleus, as you can see here. This would allow high resolution visualization of real-time growth. So the images at the bottom you can see here is basically the phyllid initiation and expansion during gametophore development. So this is the wild type moss protonemal tissue in a microfluidic chambers. So this is just, oops, so I'd been writing but didn't realize that my pen is not working. So this bottom image here is just showing you the protonemal tissue in microfluidic chambers imaged using a bright field on a wide field microscope. Well, at the top, you can see here your fluorescence imaging. So isn't it cool to actually look into how this development occurs real time, how the growth of how the phyllid initiation and expansion could be visualized in a high resolution or over real time. Another integrated PlantChips would be the detection of ethanol. Ooh, I actually have ethanol detection here. Completely forgot that I included that particular study. Oops. Okay, iPhone. So here in this paper, ethanol is detected in microdroplets by means of an enzymatic assay that converts ethanol into resorubin, or your RF. This is a highly fluorescent compound. A combination of your microdroplets, microfluidics, or your microdroplet operations, including droplet generation for cell encapsulation. And then you have the re-injection and bioinjection, facilitated the indirect detection of your ethanol via your RF, or the fluorescence of your RF in the microdroplets. So this protocol was applied to the analysis of genetically engineered cyanobacteria, just so they can distinguish which ones actually produce ethanol. So the bright field images you can see at the top shows you the microdroplets after cell encapsulation. So it's quite fascinating that you can actually, with microfluidics, you can relatively produce homogeneous droplets. So I think the diameter of this one is roughly 9 micrometers. And then you have here an image, the B image is your reinjection of microdroplets in pico-injection device after 24 hours incubation. And then your image at C is your reinjected microdroplet being pico-injected with the assay component. So remember, we want to detect the ethanol. And then for your image D, you have here microdroplets after your pico injection. So obviously the microdroplets here are a bit larger because you have now here the assay components on top of your microdroplets from the initial cell encapsulation. And if you can see the histogram, it shows the number of cells per droplet distribution after encapsulation. So here you have flow focusing microfluidic chip with flow rates of, I think, this one is 2,250 microliters per hour for the oil and for the cell suspension. So you can manipulate the flow rate for each individual channels and to control as well the size and the rate of encapsulation of your particular, in this case, cell. And then here, the other histogram shows you the fluorescence intensity distribution of your microdroplets after detection. So in orange, you have here the pre-formed droplets just containing your culture media. In blue, you have the fluorescence pattern obtained after encapsulating your wild type strain. And you know, the strain will produce ethanols. And then in green, you have the two populations profile obtained when encapsulating the ethanol producing strain. So here they use the PlantChips to essentially identify different strains for a particular application. So here, cyanobacteria for ethanol. So just imagine, I remember I designed a microfluidic device for one of my colleagues in Cambridge who was dealing with algae. So there is a way to pattern algae or also to encapsulate algae, and she's looking into the vitamin B12 production of algae. And so now the next slide. How do we branch out to active compound screening? So we have all these types of PlantChips, we have all these studies, all this developed device, all these models and designs that have been used for various applications. So how do we branch out to active compound screening? So what are the things that are actually most relevant for us in the biomedical field? So for example, in my case, I would be particularly interested in enzymes. And this is the reason why this figure on your left side, excuse me, is more on enzymes, like microfluidic device designed for enzymes. So enzymes are frequently engineered to modify their function or catalytic efficiency to increase product yield significantly through the use of directed evolution or rational protein design, for example. This requires technology like microfluidic device that could allow high throughput screening in situ. And so, like I mentioned before, microfluidics, because of its portability and flexibility, and because you can control several aspects of it, key parameters in your chip, you can couple that with anything. You can couple it with a sensor, you can couple it with your high resolution microscope or your simple confocal microscope. But at the same time, you can also incorporate other measurement technique like mass spectrometry, for example. So here, mass spectrometry is, just for those to give you a background, is a level pre-detection technique for measuring biochemical activity. And this become a method of choice for high throughput assays. Correct me if I'm wrong, but I believe this has been like a typical method of choice because of the ease and because we have a lot of mass spectrometers lying around, at least in the US and the Philippines as well. But unfortunately, this traditional type of mass spectrometry approach for screening samples are slow and require large sample volume. So for those of you chemists who have been dealing with mass spec, you know what I'm talking about. And often making them cost prohibitive because for high throughput screening using mass spec, that requires huge amount and expensive volumes of reagents. So like I mentioned before, this microfluidic device could be coupled with techniques like mass spec, and you can actually incorporate mass spec inside your device. So in fact, there are numerous examples of integrated microfluidic mass spec. There's also the electrospray ionization, or ESI, to matrix assisted laser desorption association, or what you call MALDI. Such approaches show integration of these techniques like MALDI with microfluidics. And they're very useful for both peptide and protein identification. So for most assays, these are really convenient, although of course the development of the device is quite complex and complicated. But eventually, if you manage to produce that microfluidic platform, you would have programmable control of the functions, including your droplet operations, which are likely to improve enzymatic functional screening because of electronic timing and control. So if you can control the amount, if you could use the microfluidic, or if you can digitalize your microfluidic functions, so you can control a lot of things, then you essentially could do the protein or peptide screening while, watching Netflix, because it's all programmed anyway. Okay, so going back to science, on the left side is the nanostructure initiator mass spectrometry, or they call it NIMS. It is a laser desorption ionization approach that offers very high throughput and requires very small amount of samples. That's what we want in the Philippines. The smallest amount of reagent, the better, because it takes forever to actually get reagents from abroad. And it directly measures, so NIMS directly measures the substrates and products of enzymatic reactions. And this just give you a schematic of what the name do. So essentially, the more important aspect of this looking application of what it is designed with, what source we have. So like I said, molecular classes, including metabolites and fights, in analyze and support the development of biomass degrading enzymes. Sorry, I got disconnected. Sorry about being in the Philippines. All these internet providers are scamming us all. Could you please show the slides again? Thank you. So on the left side is your NIMS, your nanostructure initiator mass spec. So it gives you an idea. This figure gives you an idea of the micro-NIMS assembly and operation. So you have there the electrodes, the chrome electrodes in particular, and the fluidic design in your A. So the image A give you an idea of of where the electrodes are. And it shows you as well the T-junction arrows and the flow, the direction of the flow. Then in image B, it gives you the array, the sealed array. So that's the B. And C just shows you the stack for holding the layers together. So in many cases, you would have to produce three-dimensional PDMS or microfluidic devices wherein you have several PDMS layers mounted on top of each other. And in this particular application, that's very obvious because you also have your electrodes there. So you have a chip containing your electrodes, dielectric, and fluidics, while you also have the chip for your drop water adapter. And for D, you have basically just here the operation workflow. So how does it injected? So in most microfluidic systems, you use the syringe pump to flow solutions or fluids. And then for F, just to show you how to mount it and the array removal. So on the right side, so the other side is another study. So on the right side was the result of novel interface coupling droplet microfluidics. So this is fascinating because I love making micro droplets. It's really therapeutic. If you've seen it under the microscope, you have your Lego-based microfluidics. You can actually do this at home. You know, if you're trying to just produce like a micro droplets of 1,000 micrometer size or 1 millimeter diameter, then you could essentially do this at home. If you, well, of course, you will need, let's say, a 3D printer, or actually, no, you can actually carve this out if you're good at carving, and then just making a droplet microfluidics. And so on the right side is that of the coupling, the droplet microfluidics, and matrix-assisted laser desorption ionization, or your MALDI, the one that I was mentioning a while ago, for label-free content analysis of single droplet. So here they use nanoliter aqueous droplets immersed in perfluorinated oil in your T-junction. So they transferred that to the capillary and then they deposited on the high density microarray and with multi-plate mounted on a motorized XYZ stage. So the figure shows basically the product of the device, wherein you have a micrograph showing the microarray with your food dye droplets deposited in vertical lines, and then your MAL-DMS imaging. And after they use, I think, BHB or your 2,5-dihydroxybenzoic acid here. And then the microarray showed you a part wherein they have the intensity of the patent blue peak. So essentially, they're just using the microfluidics, coupling it with a MALDI to look into in here they look into the droplets, the food droplets, simply the food droplets. So those are just a few of the examples of integrated plant-in chips that we could reproduce as well in the Philippines if we have a microfluidic fabrication lab. So can we actually combine plants and chips with organs and chips? Why not? You know, the question is always why not? It's not why. It's more like why not? So why not try this? And why not look into the application of, let's say, what if we encapsulate a protoplast or a certain segment of a plant? I'm not a botanist, but a certain aspect of the plant, for example, and then look into how this microdroplet with certain nutrients from that plant extracted using another microfluidic chip, and then anchor that to an organ in a chip and see how the cellular cells respond to this. So essentially, you can combine as many microfluidic devices as you want depending on your application. So if you have other questions, because I think a couple of participants last time were asking about how to contact me or where to send email if they have more questions. So here I've written my email, so I've specified there, so I know. So I'm a bit OC. So for biomedical physics work on microfluidics and nanotechnology, please contact me using my Cambridge email. And then for clinical translational research on microfluidics, so here we're looking at, let's say, drug formulation, drug discovery screening, then you can contact me using my Harvard Medical School email. And then for neuroscience and neurological research and device development, I didn't discuss much on neurological research device development, but I'm currently working on that. We're developing a new, what we call micro TMS, TMS device for epilepsy, Parkinson's, etc. And you can contact me as well in my MGH Harvard email. And then for the doctors, if we have doctor participants or clinicians, I mean, because PhDs are doctors as well. And you can communicate with me using my Beth Israel email, which is the bidmc.harvard.edu. So open for questions, and I'm sorry about my internet connection. I'm hoping to, I know, I'm not actually hoping because I cannot do anything about that at the moment. So yes, any questions?
All right. Actually, we have some questions, Jal, at the Q&A. So I'm just going to read you the first question. The first question was from Christian Mariana. It was mentioned how there are a lot of data one can get from microscope images, also optical micrographs. I would like to ask how these images are processed to get numerical data, examples such as measuring parts of the images, or some geometric information, something like that. You're Jal, I think you're mute.
Sorry, I've been speaking for quite some time.
You're mute. Okay.
So it depends on the type of image that you're looking at. So for example, if you have a contrast image, just a simple contrast image, you can use software like ImageJ to determine the pigment, the pixel in a particular area, and then you can quantify that. So that's one. And there are other softwares that could be used. For example, with my confocal microscope, one of our confocal microscope, we actually have our own software wherein we could quantify based on the intensity. So it really depends on the image and the type of microscope that you're using. So if you're looking at, there are also super resolution microscope, because I'm really fascinated with this. So I'm actually trying to find one of the images that I've seen. Actually, if I can share my background, so hopefully the internet will make it work. So, that's not the background. So this one, how do I share my video? So if you're looking at, let's see, hopefully it doesn't fail. But can you see the background or not? Because someone shared. But if you can look at the virtual background for my screen, you see there the neurons actually fluorescently labeled, and then we used film for measurement of this. So the intensity of the fluorescently labeled area is measured by pixel.
All right. Okay, we can go to the second question. It's from Janelle Saludes. So said thank you for the beautiful lecture, Dr. Fojas. Could you please provide us with further articulation on how to optimize MALDI-TOF MS and the microfluidic technique that you discussed? He said, I'm interested on how water synapse G2 MALDI DESI-TOF imaging mass spec can be used for this purpose. Thank you.
Sorry, could you repeat that? Sorry, it's a bit. Hello?
So could you please provide us with further articulation on how to optimize MALDI-TOF MS and microfluidic technique that you've discussed? There's an interest in using this for how water synapse G2 MALDI-TOF, MALDI-DESI-TOF imaging can be, it can be applied to that system.
So I've not used Synapse G2 at all, or Water Synapse G2. So I can only discuss a little bit of multi, but I am not an expert, unfortunately, when it comes to Synapse G2, which is, I think, a high definition mass spectroscopy, combining 3 dimensions of resolution, if I'm not mistaken. So I'm not an expert on this. But of course, you can miniaturize mass spectrometry, and you can look at the key parameters on how to run water synapse G2. But unfortunately, it's not my expertise.
Okay, so we have another question here from Mr. Miguel Francisco Ordonez. So what are commonly used oils and surfactants for droplet microfluidics and possibly more easily accessible alternatives? Hello, Jal. Hello, Jal. You're currently muted. Sorry.
Oops. What was the question? Sorry.
Okay. I'll be repeating the question from Mr. Miguel Francisco Ordonez. So
Yes.
what are commonly used oils and surfactants for droplet microfluidics and possibly more easily accessible alternatives?
You can use, it depends on what you're encapsulating, for example, in micro droplets. If you're encapsulating anything for that matter, but any type of oil, you can even use olive oil for, or in our case, even, do we have corn oil? Corn oil, which is more common to us. So it depends on the viscosity of the oil that you're using and what type of compound or cells or substance you're encapsulating, but you could use different oils.
Okay, thank you, Dr. Foas. We have another question from Mr. Robertson Rivera from the UP College of Pharmacy. So he would like to ask if it is feasible to do plant metabolite extraction using microfluidics.
Yes, it is feasible to do that. Yes. Simple answer is yes. Also, how complex the system would look like, we need to look into it.
Okay, so
Hello.
hello. Okay, so we have another question from Miss Mary Jane Kalagi. So she's just wondering if cytotoxicity and orthogonal assays can be done using this plant-on-chip technology.
Yes, definitely. So cytotoxicity is one of the most common applications for microfluidics. So you can put an array of hundreds of cells if you wanted to, and test different proteins, different compounds for its cytotoxicity, and then you image it. So it's one of the things that you could use for cytotoxicity. It's very common. In fact, a lot of the microdroplets or emulsion microfluidics have cytotoxicity as one of its applications.
Okay, so maybe we have other questions you want to raise from other attendees.
I'm sorry, Sir July. I think my internet is really bad. I couldn't
Okay,
hear
but,
you well.
but it seems that there are no more questions yata from our
Okay.
attendees. Okay.
Okay. Yeah.
So
Thank you,
I think
everyone.
we can wrap up. So thank you very much, Dr. Fojas, for sharing your expertise on microfluidics, particularly on this talk about plant in chip. So to our attendees today, thank you very much for attending. So this is the second of our three lecture series on microfluidics, and we hope to see you again next week, next Monday, for the third run of our. Okay, so before we end, actually, Mr. Ricardo Jose Guerrero, I would like to follow up about the surfactant question. Can you still accommodate this, Miss Dr. Pojas?
Sure. As long as I can hear you well. Let's
Okay.
see. Yeah, go ahead.
For droplet microfluidics, or unless Mr. Guerrero, can you share your question? Last follow-up question before we finally wrap up. Okay. So last question, what are the surfactants commonly used and available here?
In the Philippines, I'm not really sure. Yeah, what we could use as surfactants in the Philippines. I mean, what would be common? Because haven't tried looking into what would be the most common. I think researchers in the Philippines are more familiar with that. Baka nga Sir July or Sir Brian would be able to answer this. Yeah.
Medyo not really din, medyo not much familiar din for the surfactants din na available
Yeah,
here
in the
in our country.
Philippines, yeah.
Sorry. Yeah.
I'm not very
Okay.
familiar with that.
Okay. So
Okay. Thank you,
I
everyone.
guess that's all for today. So I would like to invite everyone for next week, particularly on next Monday, for the third lecture. So that is Systems on a Chip, Transforming Drug Development and Disease Modeling. So that is also next Monday, same time, 10 o'clock. So we will also be live on Facebook through the Department of Chemical Engineering Facebook page. So we hope to see again the attendees today, and to those who are interested in learning more about microfluidics, especially on the topic next week on systems on a chip. So we are inviting everyone to attend the talk on Monday. So I think that's all for now. So thank you very much. Thank you, Dr. Poas, and to all our attendees for today's session.